The overall goal of this procedure is to generate a purified population of human-induced pluripotent stem cell-derived cardiomyocytes. First, human-induced pluripotent stem cells are cultured in a cell monolayer until they are about 85% confluent. A small molecule compound that activates the Wnt cell signaling pathway is then added, and the cells are incubated for 2 days to induce the formation of mesoderm. Next, a small molecule compound that inhibits the Wnt cell signaling pathway is added to the cells to specify cardiac mesoderm differentiation. The cells are then incubated for 2 more days. By day 7, the differentiated cardiomyocytes beat. The heterogeneous, differentiated cell population is then incubated in a low-glucose cell culture medium to selectively purify the cardiomyocytes. Finally, the cells are visually inspected under a light microscope and analyzed by flow cytometry to confirm the production of a highly purified population of beating cardiomyocytes.
The main advantage of this technique over other cardiomyocyte differentiation protocol that utilizes small molecule is that it employs an additional glucose deprivation step. This enables us to purify cardiomyocytes, which are able to utilize sources of cellular energy other than glucose.
Non-cardiomyocytes can be selectively eliminated from this heterogeneous population of differentiated cells. Cardiomyocytes can then be utilized for downstream purposes such as in vitro disease modeling or high-throughput drug discovery assays.
Prepare for